is Konstantin Mischaikow as this week's camp Colloquium speaker. Konstantin was a student of Charles Conley, who was one of the main figures in dynamical systems in this country during the 20th century. He had been a student of Jürgen Moser, and when I was a graduate student at Berkeley in the 1960s, there were sort of three major figures in dynamical systems in this country. My thesis advisor, Steve Smale, was one, Jürgen Moser was the second, and Jack Hale, who was at Brown at the time, was the third. And Charlie was sort of the singular representative of the next generation, as it were. And Charlie's particular interest was in using topological methods in order to study qualitative properties of dynamical systems. He created this whole theory that is a topological theory of how to decompose dynamical systems. And Konstantin has been the foremost proponent and inheritor of that work. And I think what he's going to tell us today relies upon that a great deal. Thank you for coming from Rutgers, and we're delighted to have you.
It's always a pleasure. So, there's not a typo, that is the title. And so, let me give you an explanation of where I'm going with this. Let's make sure it works. So, let's start at a level of modeling. There's a nice picture of the solar system. I didn't take it, I stole it from Google. But if I asked you for a model of this, I don't think anybody would really get object if I wrote down these differential equations. If I want to do something with these differential equations, okay, I've got to get somebody to go out there and weigh each of the planets so that I know what masses to put in there. And I should get somebody to figure out the initial conditions, where are the planets actually, how fast are they moving. But if I have that initial condition and I start solving the differential equation, at least the first order, we're probably going to do a pretty good job of predicting what's happening. So let's start with a different picture. Let's start with that picture. That's a picture of the EMT network, epithelial mesenchymal transition network. It's in us. It controls how well cells stick together. The epithelial state means that the cells are well stuck together. The mesenchymal state, the cells separate and they move around. Why do we have this? Well, if you cut yourself, then you want cells to migrate so that they hook up to each other, and then you want them to reattach to each other, and that are different stationary states of this this network. All right? That's clearly a dynamic process, right? Something told it to turn on and change to a different equilibrium state. So we should write down some differential equations for it. And that's about the best I can do. Right? Why? Well, I mean, this is relatively simple. I mean, okay, it takes Newton to come up with it, but what's really going on here for every one of those arrows is that you've got translation going on, or transcription going on, making mRNA. The mRNA gets translated into proteins. The proteins have to come back, and you're going to do this regulation, and they have to bind to the DNA, and all these are fairly complicated chemical processes. There's what nonlinearity describes this, and then what are the set of parameters that, the masses that you've got, what should be put in here. And then finally, If you're me, there's only one solar system. I guess if you're Kate, then there's lots of solar, or Alice, then there's lots of solar systems out there, right? But the lots is very relevant. You know, we think about trying to deal with what is going to be the fate of one of our cells under this organism, under this dynamics. There's lots and lots of cells in our body, and they all have slightly different initial conditions. They all have slightly different mutations. They all have slightly different epigenetics. So even the initial condition is not something that's going to be determined. Right? But I'd like to do this. I'd like to solve this dynamical problem. Okay, so that's what this lecture is about. How can we go about trying to do that in a mathematically defensible way? Now, what I'd really like to talk to you about is some of the work that we've done on this. But I only was given an hour. I'd also like to talk to you a little bit about this. This is P53, where a key feature that I'm going to talk about here would be oscillations. This is the well, it's the RBE2F pathway. This is the switch that decides whether a mammalian cell is going to flip or not, reproduce or not. So this is really a switch. Oscillation switches, I think of switches in terms of equilibrium. Again, we've done some work on that, and I will kind of result drop along the talk. But these are too complicated to get the essential mathematical ideas across. So I'm going to go to something really, really simple, and that's the genetic toggle switch. And this is one of the first synthetic networks that was developed. And I'm going to simplify it to a picture like the picture I started the talk with. You've got two genes, and they interact with each other by repressing each other. So the whole talk is going to be focused on trying to tell you how I would like to solve this differential equation. And that is the differential equation. That's the model, and that's what I need to solve the dynamics. Now, I also like to pretend I'm an applied mathematician, and so I would like to think that what I'm doing is useful, and if it's going to be useful, I've got to be able to compute. All right, so there's two things I really want to talk about. One is computability, and two is content, and this is to try to convince you that the computations I'm doing actually have some meaning for dynamics. And I could have put it either way. I'm going to do the computability first, so for those of you who like proofs and content to the talk, please bear with me. But I'm going to try to show you that after you've put all this together, there's a way to interpret what has been done in a meaningful way. All right, so that's my goal. I've really tried to strip it down to the absolute minimum because I'd like to communicate the ideas to you. And so if you have any questions, please stop me. I mean, I'd rather not finish the talk than have all of you wander away and shake your heads. You might do that anyway, but at least if I communicated the ideas, that's fair. All right, so let's start with a mathematical interpretation. This is my model. That's what the biologist gives me, and that's where I have to start. So the assumption I'm making, and this is a really strong assumption, but the assumption I'm making is that the dynamics that I'm going to see from this can be represented by a differential equation. I have no idea what that differential equation is, but I am assuming that it can be represented by such. There's two things going on. I'm going to let xi denote those quantities. If we think in terms of a gene regulatory network, you can think of these in terms of the quantities of proteins that are there. You know, this is my model. So if I'm going to try to tell you something about the fine structure of this from that, you should all leave, right? Because I'm not giving you very much, so I'm not going to ask very much. My notion of dynamics at the moment is going to be, is x going up or is it going down? And in this context, it seems reasonable to say, well, these are, let's say, proteins. Proteins probably have some kind of degradation rate, so there's some degradation that's going on. And they're being produced by genes, so there's some production rate. And that's kind of my model. I'm very consciously at this point not writing dx dt here. I think if you put a dx dt at this point, you're misleading yourself. This is supposed to be a representation of the dynamics. It's not supposed to be a differential equation. So for the purpose of this talk, I'm going to just assume that this quantity, the degradation rate, is a constant, that it's not affected by anything else. We've started modeling with this more complicated term, but that requires more modeling assumptions. So that's what I'm asking. Is xi increasing or is it decreasing? That's the fundamental question. And what I'm going to do now is to come up with, again, a representation of lambda. I'm not writing down a differential equation to be solved. I'm trying to get a representation of the dynamics. Okay, and I'd like to do it algebraically. So I look at this, and my assumption, and I haven't had too much feedback, pushback from biologists when I tell them this is how I'm interpreting their diagram, is that x1 represses the production of x2, and so if I think of what that might mean, it is that if I look at the rate of production of x2 when there's a little bit of x1, then that should be higher than the rate of production of x2 when there's a lot of x1. Do I know what those rates are? Absolutely not. So let's give them names. So this is the rate of production at the low level, and then there's some difference between low and high. It should also kind of give you a sense of where low is happening and where high is happening, so I'll give it that another name. And if that's all the information I'm giving and I want to have a simple way to calculate on that, I'll use a step function. But before you fix on any particular step function, I do not know L, I do not know delta, I do not know theta. So in particular, if we're looking at this picture should be valid for any theta in between. So I'm going to solve this problem for all the L's, deltas, and thetas. Right? So don't think of this as a hard fixed step function. This is just a calculating tool, but I'm going to calculate over all possibilities. Okay, let's do some algebra. This is my level of algebra. If x1 is less than this threshold, wherever it might be, then x2 is going to decrease if this quantity is negative, right? I mean, my model is decay versus growth, and so if the decay overwhelms the growth, so if that's negative, then we have decrease. Another way to write that would be to write it this way. It's simple, all right? If x is bigger than theta 2, 1, well then I'm in this regime, and so this is the inequality that I care about in terms of growth or decay, right? Okay, now I'm going to get very fancy, and I'm going to switch to Mike's language. This is a semi-algebraic set. Right? So a semi-algebraic set is just some set that can be written in terms of inequalities of polynomials. I'm also writing it down over here as a partial order. This semi-algebraic set notation is actually very important because this is trivial, but in general, I think of these growth rates, if you have a complicated network, as being given by sums and products of these kinds of representations. And in the end, I'm still interested in things increasing or decreasing, and then these sets are not so simple. And trying to characterize all those sets is hard. Right? That's, and if you do it naively, you run into, the problem is always solvable, but it becomes devilly exponential. So we've been spending a lot of time trying to solve these kinds of problems. Right? And you will see that I talk a lot about partial order. So those two concepts actually are going to play an important role. So in summary, what I've done is I've gone from this picture to an algebraic device that I'm going to use to compute, and a parameter space which consists of 3 parameters for every edge and 1 parameter for every node. There's a k rate for every node, and for every edge there's a low expression level, a higher expression level, and a place where that expression level changes. And I'm going to care about the signs of these things. So, again, what am I really talking about? What can I get my hands on? In terms of these parameters, the only parameter that I've put in there that is actually a quantity is the theta. Right? The gammas, the L's, and the deltas are all rates. And so if I want to talk about x, then the only thing I have at my disposal is theta. And so really what I'm going to do is I'm going to go back and I'm going to look at the sign of this thing. I'll try to convince you that that really makes sense, to think about it that way. All right? All right, so if I write it down this way, then you can see now that there are 3 possibilities for the sign of this. Again, I don't care about the fine stuff if x is not changing. That's just too improbable, all right? So I care about inequalities. And there's 3 ways these inequalities that I can arrange these quantities. This can be less than L1, which is obviously less than L21 plus delta 21, because this is assumed to be positive, right? It's the positive change from the low to the high expression rate. Here's another inequality that's possible, and there's another inequality that's possible. I want to organize these. If I have a higher dimensional problem where there's lots of nodes coming in, I'm going to get lots of possible inequalities. So the way I'm going to organize this is as a graph. I'm calling this a factor graph, which will lead to a parameter graph. So there's 3 nodes in this graph for these semi-algebraic regions of my parameter space. They're in this case polytopes, but let's call them semi-algebraic because that's what happens in general. And then there are edges between these nodes, and associated to those edges is a place where there's an equality. Right? So these are regions, and these are saying that there's a hyper surface between them. So notice I've given you a semi-algebraic decomposition of the positive orthon in 4-dimensional space. Because I have 4 parameters for that edge, and here's an explicit decomposition. It's kind of hard to work because I really do care about the structure of parameter space. So let me just say at the moment that there's a natural homotopy for this problem, and it extends in a natural way, to setting all the gammas equal to 1 and putting it on the unit simplex. And this homotopy preserves these semi-algebraic sets. Yes?
It looks to me like there's kind of a logical structure in this graph of possibilities where gamma 2, theta 1, 2, relative to the other quantities is kind of moving upwards as you move down the graph.
Yes.
Are there situations in more complicated problems where like the structure of the graph is like not obvious or not possible to create?
Oh,
because you have a complicated...
We always create this.
Okay.
And that's
It's always
part of
possible.
why we can do the computations. But the structure of the graphs gets complicated. Gets very complicated. Right? Yeah. And these graphs get very big.
Right, I'd imagine.
Okay, so I'm going to draw you a picture now of this representation of parameter space. So gamma 2 is set equal to 1, and I told you that I'm on the unit simplex, and I gave you the inequalities. I've now said gamma equal to 1, right? And so I can go through and I can actually see what these look like in this simplex, right? And it's really kind of nice. I can get my hands on it. Now, delta went all the way down to 0 here, but this is supposed to measure the difference between a low expression rate and a high expression rate. So setting it equal to 0 just doesn't make sense. All right? If it was really equal to 0, then the impact from 1 to 2 never changes, so let's just take that edge away. Right? So let's move up a little bit. The same thing is happening with respect to this threshold, right? The threshold is where you change from low to high. If the threshold was 0, then I also wouldn't see any changes. And then just for a technical reason, let me also do the same thing and the L's are always non-zero. Okay? So here is a polygonal decomposition. Right? And if you're an algebraic topologist, that's a CW decomposition. This is the kind of object that I can start doing algebraic topology. Okay? So parameter space is now given in terms of a nice, at least combinatorial descriptor that I can do, tell you about the topology of, or at least the algebraic topology. Okay. So let's go to the full network now, right? What I told you about was this, but there's also this edge. And if you look at the parameters that are involved, those parameters don't overlap at all. And so the full parameter space, which is 8-dimensional, is actually a product of these two 4-dimensional parameter spaces. And I told you how to decompose this node, the parameters associated with that node. Here's, well, this one looks exactly the same, so you get the same factor graph there, and the full parameter space is a product of these. So this parameter graph is, keep referring to it as a parameter graph, that is how I organize parameter space. And this region right here is given explicitly in R8 by this set of inequalities. Now, of course, I'd like to do dynamics. So the next thing I have to do is to figure out what's the dynamics that's associated with each one of these regions. Okay, so let's represent the dynamics. Again, I told you that the only parameters that I see in my phase space are where this function theta changes. This is how I keep the function lambda changes. This is how I keep the difference between the high expression levels and the low expression levels. Right? So if you pick a point in parameter space, you naturally have this decomposition of phase space. So let's go into one of these regions. Let me write down the equations first. Those are the equations we're looking at. What happens if you set the x is equal to 0, so you're along these? Well, and this was why I wanted to make the L's positive. It just makes life easier for me. Then the arrows are all pointing in. And if you go out to infinity, then this clearly dominates, and so everything's pointing in. So all the dynamics is taking place in some compact region in the interior. Let's pick one of those parameter regions. This is the one where, well, what's happening here is I'm telling you decay is dominating over everything else. The U's are the L plus deltas. All right? It's easier to write it that way. So decay is dominating. So what has to happen? No matter where you are, you have to see x1 go down and you have to see x2 go down. Well, that's a representation of that. We call that a Rook field. So that's a representation at this region of parameter space of what's happening to these regions. So let's try to give a nice combinatorial representation of that dynamics. So to each region, I'm going to associate a node. I'm building a new graph. And what happens when I go to this space? Well, this vector, this field is moving to the left. This field is moving to the left. So I should go take this region and map it into that region. And so I'll do that across every one of the faces, and I'll see how things go. So for those of you who've heard about Boolean networks and things like that as representations for systems biology type problems, you'll know about state transition graphs. So we've just recovered a state transition graph, right? So these are these edges that I found by comparing boundaries to the Rook field. And notice all the arrows are pointing in on this edge, so I'll put a self-loop there. And that's a statement about combinatorial statement of dynamics. Yes?
I was wondering, going back to the kind of the choice of looking at the thetas and putting them in for x, can we know, certainly it makes sense like maybe then on the boundaries, is that going to, by plugging in theta for x to get the decay rate, that allows you to draw the arrow at the boundary, even though maybe not in the interior of one of the boxes, you no
So
longer.
in the interior of the box is what I really know what things are happening. And when I get to the boundaries, I might, this is a nice simple case, I'll show you exactly a place where we get an ambiguity at the boundary, okay? But for the moment, let's just pretend I can do this.
Okay.
Okay?
Yeah.
All right. Yeah.
Is there any reason that you don't put an edge from the source node to the sink node in the sweep transition graph?
I want to do my computations as simply as possible, and so I'm just looking at the face relations. Right? I get worried that maybe something less subtle will happen here, and I will show you very quickly an example of it, okay, where everything gets subtle and a little bit there. All right, so that's my state transition graph. Even this is much more complicated than I think is necessary, as John referred to. Advisor was Conley, and one of his major results was this decomposition theorem that decomposes dynamics into the recurrent parts and the non-recurrent parts. And so I'd like to do the same thing here. I'd like to identify what parts are recurrent and what parts are non-recurrent. And so, well, it's a graph. There are very, very quick algorithms for taking a directed graph and looking for the recurrent components. So if I look at these will all be strongly connected components, if you know what I'm talking about in terms of topological sort. But this is a special strongly connected component because it actually has recurrence in it from the original graph. And so what I'm going to do is I'm going to go to a graph like this. I'm going to look at the strongly connected components that actually have recurrence. And then I'll make a new directed graph where I only have those things as nodes. And I'll use the ordering from the topological sort to tell me how those recurrent nodes fit together. Okay, so in this case, there's only one recurrent node. That's that recurrent node right there. All right, so that thing, that's all I'm going to store. Everything in here kind of wanders down to there. That's the idea on this combinatorial level. Oh, I missed something. Sorry. You see, okay, I found this. Does this have any dynamic meaning? If you go back and think about the dynamics, this is a region where everything's mapping in. So this has actually identified the fact that if these arrows have anything to do with some kind of differential equation, then there will have to be some invariant set right there. Okay, that's compact region. It's getting mapped inside itself. So nested sequence of compact sets are non-empty, so something's in there. All right. Now we get to something a little bit dicier. All right. So here the root at this place, the root field is pointing this way and it's pointing that way. Again, I'm only going to look at what happens across faces. There's my state transition graph. This is the arrows that come across here and here. Here there's a self arrow. Do the same thing. I find this strongly connected components. I keep track of which ones are the recurrent components. And so my Morse graph is going to, once again, is in this case going to have 2 dots to it, right? This is a directed acyclic graph. It's a poset. Right? It looks very fancy to be talking about that here. I just convinced that's really important, right? And so in the phase space, something happens here, something happens there. Well, let me just, as a mnemonic, we'll put some red dots in there to keep track of what's going on. Right? So I told you how I'm going to compute dynamics. We'll get to the content part later. What I'm trying to convince you is that I can do computations extremely fast. So here's my database, the parameter graph, and what I'm going to fill it in with is the dynamics that I've just computed. Right? So that was that single Morse graph, that first computation where the gammas dominated. This was the second one where the gammas were in the middle. Go through and do all the computations, that's what you're going to get. And here's where those recurrent sets actually reside in the phase space. So all of this information is what we can store. All right? And that's a very small amount that has to be stored relative to all the possibilities that are out there. Okay? So this is the database that we compute. It's purely combinatorial, both in terms of the decomposition of phase space, the parameter space, and in terms of description of the global dynamics. So that's what I call the database. You can look up the dynamics for any possible parameter. Like I told you, I was going to kind of drop this in to convince you that maybe we can actually use this for interesting problems. So here's a subnetwork of P53. It's this part right here. Like I told you, people are interested in oscillation for this system. The parameter space is 29-dimensional, 3 times the number of edges plus the number of nodes. The number of nodes in the parameter graph is 800,000. You input this into your laptop, and 37 seconds later, you have a complete database. The point is, these computations are extremely fast. Okay, so now I have my database, and I can go back and I can say, well, these guys care about oscillation. Does oscillation happen for this network, and at what parameters? Well, if you're going to see oscillation, it should be a minimal node of this Morse graph. And there should actually be some recurrence going on. It should have come from a place where edges were, where you had cells mapping to cells back in the phase space. So we query for that, and there's 7,000 of those nodes in which that happens. Okay, so this is all can be done within a minute. Then this is now explicit regions in parameter space where this oscillatory dynamics is taking place. I claim that even at this level, we can do some data analysis. So here's a hypothetical, because real data is just too messy. So here's a hypothetical example. And this is actually taken from what Collins did in that toggle switch paper. So the idea is that I've got 2 genes that I could measure their expression level of, and I've got some chemical I can pour into the cell, and what that chemical does is it increases the degradation rate of product of gene 1. And as I change S, this is the outcomes that I measure. I've got 2 genes. They interact somehow. Well, let's write down models for it. Right? Those are the kinds of models you can see. In fact, there's some more. There should be the possibility for self-activation or self-repression. That's just going to messy the water. So let me not even think about those at the moment. So let's just do this. For each one of these models, I can give you a database. It'll take me about instantaneous for something of this size. So, and the idea is that I'm going to be pouring that chemical in.
Sorry, Konstantin, I find myself lost. Could you explain the colors and the...
Yeah, so I have 2
axis,
genes.
please?
So this is, I have, yes, here, this is green, green and red, which is probably not the best choice of colors for some people. So here's green, and so I'm seeing the green expression, right? So I pick an S, that's the amount of chemical I pour in, and then I do a measurement of the expression levels of the gene. And here was what I saw at this measurement. I saw sometimes in some cells there were low red and high red, other cells there were high red, low green, and also high green. But when there was very little S, then I consistently saw low green and high red.
So for each experiment, there are 2 dots on the screen here, 1 red, 1 green.
Yes,
correct?
yes.
Okay. And when you are showing the graphs with arrows versus those other connectors, this is repression versus...
Right, so I'm asking, if I made this as a model, could this be consistent with that expression?
So there's this code that biologists use where a thing that looks like this means if I'm high, you're low.
Right.
So that's, maybe they don't know that notation. The arrow means I'm on, that fires you up to be
Yes, right.
So that's
Okay, yes, yes.
And S pointing into node 1.
So this is this chemical I'm going to dump in.
But why?
Oh, because the assumption is that S increases the degradation rate of product.
Got it. Thank you.
Okay. Other questions? Oh, you're just raising your hand. Okay. Okay. So I take this network and I compute the database, which is, in this case, instantaneous. All right, let's compare this to data. If I have low S, right, increasing S means I'm moving this way. I'm making gamma 1 larger. this is what measures the red, and this is what measures the green. At low S, green is supposed to be low and red is supposed to be high. Well, this is red high, green high, so this won't work. But here I have red low, red high, green low, red high, green low. Oh, so maybe I'm in one of these settings. But on the other end, I've got to have low red and high green. That fails here, so this must be the only swipe that is reasonable. And not only that, but if you do that, in the middle, you should see low, high red, low green, and low red, high green. Hey, that's what I see. Okay? So this is consistent with the data. If you try to do it with either of these models, you just can't find anything that's consistent as you march through.
Sorry, I'm still
Yeah.
trying to map two pictures together.
Okay.
So in your diagrams, the horizontal coordinate is red, and
This, yes.
and
Yes,
I
this
see.
is red, and that's green.
And that one is green. Got it.
And there are, of course, no numbers, right? I mean, it's just relative quantities, right? So have I found the right model? No, I'm a mathematician. This is a biology problem. What I can do is rule out some possible models, and maybe if it was more complicated, I could suggest a set of experiments that should be done to verify this, all right? For example, you could go in and say, well, gee, if this is really the model, then I should try to do the same experiment by changing the degradation rate of gene 2, or by upgrading the expression rate of gene 1. Right? So that I can predict, I can say, I can give something predictive that should have qualitative differences. Yeah.
I'm still confused about that, those arrows.
These.
No, the one, two, like, what are the arrows?
Inhibition versus
Oh,
activation.
this says if this is on, this goes off. And if this is on, this goes off. Here, if this is on, it turns this gene on. Okay?
So that second one turns on, two turns on.
Off. Yes.
And two turns off.
Well, here you might see oscillatory behavior. You won't officially, but you might see oscillatory behavior in something like that.
Can I ask you a sociological
Yeah.
question? So this way of speaking, where things are on or off, or this, like a biologist, if there were some in the room, would say this upregulates that, or it seems like it's really nicely matched to combinatorial thinking, to topological thought. Like, people who are in the world of estimating parameters of all, that's sort of usually too specific for a typical biologist, or they say, I can do those experiments. Did you come to this way of thinking because it's topology, or were you hanging around a lot in biology labs?
So about 10 years ago, a little bit longer, I really actually took things like the lac operon and the phage lambda and said, I'm going to be a real applied mathematician for once in my life, and I'm going to find parameters.
Yeah.
And we worked really hard. So this is work with Tomaskevich. And we worked really hard. We scoured the literature. We went after parameter rates. We wrote down the models. We looked at a bunch of models. And every time we tried to fit parameters, what we would find is that you get these huge regions of parameter space where basically you were getting the same results.
Mm-hmm.
And there was a talk by Odell at one of the Cyan conferences. And he was saying, this is what biology is all about. And my reaction was, well, how do we do math where we don't see bifurcations?
Gary Odell is saying it's all about getting parameters, or
No,
the
no.
sloppiness of the parameters.
It's the sloppiness of the parameters, right? And so he was saying, I have, this network, and he made up a network based on polarity segmentation, whatever it's called, right, for the fruit fly.
Yeah.
And then he said, basically, it doesn't matter what parameters I put in, I still get this pattern out. And that was kind of like the opposite of what I learned about bifurcation analysis, right?
Yeah.
Which is you do something and things change. And here, how do I find the places where there's no bifurcations,
Uh-huh.
or at least that I can see nearby, right? Okay, so that's what I'm trying to do here.
Mhm, that's nice.
Okay? All right. Let's talk about it in this context. This is a serious problem now. So what Yao did here, because, okay, so we're here at this model. The actual, what you should do, like I do, Google RBE2F network, and you'll get a full page with minuscule little dots and lots of arrows. So then after I don't know, a half dozen papers and high-profile journals, you end up with a picture like this that's put together by you at Duke. And their question was, gee, this is too complicated for me to analyze. Could I make a simpler model here that would still act like a switch? Because this is RBE2F. This is what decides whether a mammalian cell replicates or not. Obviously, it's very interesting because this thing, when it fails, is really related to cancer. Okay? So he proposed that break it up into 3 groups. Here are the edges between those groups. Those are what the biologists think really happened. So can I make a model with 3 nodes that consists of some of these edges? And if I'm making it 3 nodes, which edges are really important? And so what they did was they wrote down a bunch of ODE models, and then they randomly sampled parameters. So they fix a subset of edges, write down the ODE model, randomly choose parameters, and then see whether that thing behaved as a switch. Just kind of like I did for the toggle switch. You take one parameter, that's this one up here, you push it along and you see whether you see a hysteretic type phenomenon. It's a very nice paper, it motivated our work. My take on this today is, no, that's the wrong thing to do. What I think you should do is you should consider taking this thing and subdividing it into, and representing your model as a model with 5 nodes. And here are the 5 ways you should break it up. Why? Because if I break it up that way and I look at all possible different networks with respect to edges, and I now ask how many times you fix one of those networks, how many times when I sweep across do I see hysteresis? With this particular network, I get it about 13% of the time. That's the highest that we could get by looking at these kinds of models. Okay, I can do that computation. Doesn't mean anything. Well, I'm not an experimentalist. But I can go, and actually Steve Haas is the one who said, oh, I've seen that network before, because this is the yeast start network. This performs exactly the same function for yeast as for humans. All right, and that sure looks very similar. Okay, so that at least suggests we're finding something that's not completely nonsensical. Now, I left this out, P20. Yeah.
I was just wondering if I understood you correctly when you were sort of the takeaway of like, we're saying that the 13%, that's actually pretty good, that's high, and that you tried a lot of different subdivisions, and you tried kind of, okay, what happens when I forgot what's changing here for the hysteresis.
It would be this, the growth signal here. So.
Okay, the growth signal.
Yeah.
But so you're saying, okay, there's all these different ways we can subdivide them, but which sort of give the biggest swath of parameter space behaving the way that we expect it to.
Yes. And we didn't do quite as extensive of what you're giving us credit for, because we kind of started with theirs and then said, what happens if we make it bigger? And so I think we should redo it by as extensively as you said.
But yeah, but considering a lot of different
Yeah,
possibilities.
yes, lots of possibilities.
Yeah.
Okay, I think that P27 should definitely not be put into that node, even if you want to go down to 3. Why? Because if you take P27 and now add it to this network, you see suddenly you drop from 13% to 0.35%. I think of that as a real victory, because P27 is actually one of these regulatory genes that gets in the way of reproduction of cells. It senses things from outside that suggest that the reproduction of the cell is going bad, and it slows things down. So its whole purpose is to break up the switch behavior. And we see that extremely clearly from these percentages. Now, again, doing experiments in human cells is a little bit complicated, but we're trying to do this kind of stuff with Steve Haas on the yeast cell site. I've got it right here. What about parameter space? I mean, that's my big deal, is that we're actually telling you about huge swaths of parameter. So here's my parameter, here's my database. I pick a phenotype, for example, bistability, that's happening, that's happening right here. What's the topology of that set of parameter space? Oh, that's going to be the product of green by green, right? It's this region product with that region, and that's clearly a nice polytope, and that's clearly contractible. Okay. What about where you have monostability? Well, here's my CW complex, it's a product, but I can go and tell you which are all the cells. It will be all the cells except the green cross the green.
Sorry,
Yes.
in the spirit of really defining things, what is contractible and what are the implications?
Okay, contractible really means that I can have a homotopy that takes that cell and contracts it to a point. Okay. Implication is that I can get from, anytime you see bistability here, I can get from one point of bistability to another point of bistability without losing bistability. Okay, it's possible. Okay. But let's look at this. I mean, from the picture, it looks like it should be a circle, but we're really in an 8-dimensional space. All right. I've done a homotopy to this for this, I did a homotopy to this 2-dimensional space. So the topology is being preserved. So it's easier to compute homology in a 2-dimensional space than it is in a 4-dimensional space. I'm now getting computing homology in a 4-dimensional space rather than an 8-dimensional space, but I'm preserving the topological features. And there the answer is yes. This really does look like a circle. Right? At least up to homology now. I'm not claiming homotopy, but up to homology. Right? So we can start doing these kinds of calculations and get something about the structure of parameter spaces that exhibit a phenotype. Where might this be useful? Well, you know, if you are here and you want to get there, in terms of a phenotype, you should try to run your drug scheme this way rather than try to run it that way. Right? That's, if I'm thinking about controls now, knowing homotopy types becomes very important. Can we do that here? So now I'm in a 29-dimensional space. Of course, I've done some collapses, but yeah, we can compute the homology in this setting, and so we can prove that the region of oscillation is again contractible. Right? So we can actually start working with reasonable networks and tell something about the geometry of.
Sorry, does that mean you can move among those 7,000
Yes.
nodes and it's
Yes.
they're all connected?
They're all connected, and not only that, but it's contractible. I mean, it's not a donut or anything like that. Okay. Okay. You've been patient and listened to me tell you how to compute. Am I doing any mathematics? That's what I want to talk about. Now, I need to introduce a little bit of technical stuff before I tell you that I've actually solved differential equations. First one is, well, everybody knows what a poset is. If you give me a poset, I can always make a lattice. It's going to be the lattice of downsets. So you just take an element of the poset and you look at everything that lies below it, you make a set out of that, and now I've got a lattice under inclusion. Similarly, if you start with a lattice, finite bounded distributive lattice, then, well, like this one where it's ordered by inclusion, you can go and look for those elements in the lattice that have a unique immediate predecessor. That's what these elements are. I've given you an order relation, so this now becomes a poset, and gee, this poset is isomorphic to that poset. That's not a mistake, that's not a lucky coincidence, that's Birkhoff's theorem. Right? So posets and lattices contain, at least on this level, finite, bounded, distributive, that kind of adjectives, contain exactly the same information. This clearly is smaller. This is larger. I want to compute here, but this is where I'm going to see topology. This is where I'm going to see dynamics. I told you about topology. I want to compute topology. To compute topology, the nice way to do it is to work with cell complexes. They are exactly what you think of in terms of triangulations, all right? I'm going to think of it as a poset where you have face relations. You know, if you have a triangle, then this is bigger than the face that's associated to it. And this is the conditions you need in order to be able to do algebraic topology. Yes, so this is that fact, that if you have a cell complex like this, then I can make a chain complex so I can start doing algebraic topology. If I have, right, this is a poset. If I look at a down set of the poset, that also turns out to be a cell complex, and so I can set up a chain complex for that. So I've got a lattice structure here, and now I suddenly switched lattice structures on you because I want to do algebra rather than combinatorics. And there, the lattice operations, instead of being intersection and union, they are, I have vector spaces, for example, so it's intersection of two vector spaces, or the span of two vector spaces. Those are my two lattice operations. One more definition. If I have a lattice filtered cell complex, so that's going to be one of these cell complexes together with a finite distributive lattice and a lattice homomorphism between this lattice and this lattice of subchain complexes. That's the technical stuff. All right, let's solve differential equations now. Oh, example. If I had this order, then here would be what Birkhoff would tell me to look at that lattice. Here's a complex, cell complex, and this is now a lattice filtered cell complex. Here's my lattice, and there's the filter. Okay, so these are fancy words for actually pretty natural things. Characterizing dynamics. This is where life is bad. Why? I mean, these arrows don't point to another cell, right? And that's why I'm trying not to go across edges, lower dimensional edges and all this kind of stuff. What's clearly here is that I'm missing a separatrix for the dynamics. Why am I missing it? It's because my cell complex that I've made, this cubical complex, is just too small. It doesn't have enough room for me to see the algebraic topology. So, if I have this vertex, these edges, this two-cell, this vertex is a face of this is a face of that. These are combinatorial representations, and I can make a new complex where if I look at any face relation in here, it becomes a cell in this new complex. That's what it looks like. The details aren't so important. What I'm telling you is there's a purely combinatorial way to go from the kinds of calculations that we've been doing to this larger cell complex. Okay? I'm drawing pictures, but really everything I'm doing is combinatorics. I'm going to draw pictures of vector fields crossing this, but since everything is combinatorial, there's no reason to think about straight lines. These are CW complexes. If I was a great geometer, then I would know when you gave me a differential equation, I would say, here's the combinatorics, and I'm going to embed it into phase space in such a way that everything I'm telling you is correct. All right? I wish I could. I want to transfer what I call a transversality model for dynamics. So let's start with this cell complex, this I call face complex. And for every single top-dimensional cell, let's put a self-edge. And for every codimensional one face, let's put arrows in both directions. Let me do what I've done before, which is I write down the state transition graph. That's the state transition graph. And I look for I do a topological sort, and I look for the chain recurrence set. Everything's recurrent. So there's my Morse graph. What's the dynamic meaning of that? Well, there's something in there. Okay? So I have just achieved, the ultimate mathematical stereotypical result. It's absolutely correct. Pretty useless. So let's do something a little bit more useful. Okay? So there I am. Now, what I want to do is I want to remove any edge that's not consistent with my rook field. Now the rook field is pointing this way, and it's pointing that way, so it should be pointing that way across that face. Or, sorry, don't look at the face. Don't look at the face. I have to keep telling you. Right? This edge, I think of as somewhere over here. The rook field tells me I'm going in that direction across the edge. So get rid of the arrow that was pointing the other direction. do that systematically. Right over here, this is an edge sitting in here. The arrow's pointing that way. There's no reason to have an edge there. So do that consistently. And now, there's the state transition graph. There's the Morse graph. I'm looking at the recurrent components. Oh, suddenly I have an interest in Morse graph. Right? That's my POSET.
Sorry, how are self-transitions, let's say E, which you had in the previous slide, how are they consistent with your Rook field?
The E, well, they don't contradict it. I don't ever want to lie to you, right? I don't really know what's happening in here. So put an edge, a self-edge. I haven't lied. I might be too conservative, but at least I'm being safe.
Okay.
Okay? If I give you a POSET, or give me a POSET, I'm not imaginative, Birkhoff tells me what to do. Birkhoff says, look at the lattice of downsets. Right? And within the lattice of downsets, the important things to look at are the joint irreducible elements. Now let's go back to the phase space. I'm going to build you one of these lattice morphisms. So what I do is, I go down here and I say, well, gee, this is something I should look at. Where did A come from? A was something, let me check my, what did I just do? What's the wrong button? So A was up in the upper left-hand corner. That was this, this region in the upper left-hand corner was a cell that contained this, right? B is sitting down there. C is this one, it's A, B, and something else. There's A, B, C, D, and there's A, B, C, E. So these are now subsets of phase space. These are all attracting. These are, everything's pointing down here, and if you check with the transversality conditions, these are all the transversality conditions. They're pointing in a certain direction because I've sorted out the recurrent parts. Right? These are locating all the recurrent parts. So this is a lattice of attracting blocks. If I go and look at this guy, which is the unique, this is a joint irreducible element, so it has a unique predecessor. Let's look at the topology of this guy relative to that guy. The whole thing is an attracting block. These are attracting blocks, so anything that goes from this region into that can't get out again. So what I've just found is an index pair, and I can use that to compute the Conley index. Okay, why do I get excited about that? First thing is, that's all combinatorial. I gave you the pictures over here, but it's all combinatorial. If you take this and do a different embedding into this space while preserving this combinatorics, I've already done all the algebraic topological computation for you. So Conley theory is great because it tells you all about dynamics. All sorts of different kinds of dynamics. Algebraic topology, which is the Conley index, plus something, and all these plus somethings can be found from the dynamics that we've, this combinatorial dynamics that we've computed, gives me things about dynamics. So what I have now is this big complex. I want things to be simple. This is too complicated. Is there a simpler representation of this object that's still going to contain all this information about who gets connected to whom? The answer is yes. There's something called the connection matrix. This was Conley's idea and carried out by Bob Francois in his thesis originally. Kelly Spenlove has just redone this in a combinatorial setting, and it's much cleaner. We can now do these computations, and in this context, what that is, well, this homology, if I did this relative its immediate predecessor, it would be boring homology. So there's no interesting algebraic topology here, no interesting algebraic topology here. This gives me algebraic topology on the first level of homology. This gives me algebraic topology on the 0th level of homology. I'm making a new chain complex that where the boundary operator says this guy is mapping to here and this guy is mapping to there. Right, so out of this, I'm going to extract just this little piece of information, It's telling me about global dynamics. I have Conley index information here, here, here. Previous slide was to tell you that that's useful information. I have connecting orbit information, so I actually understand something about the gradient-like structure. Dynamical interpretation of this picture is there's 3 invariant sets, and this one connects to those 2. So there's my new database, where instead of carrying around all these pictures, I'm now carrying around the algebraic topology. These are all chain complexes. If you got chain complexes and one is sitting next to the other, you would say, how do I transform from one to the other? Well, you need chain maps for that. All right, and those are called transition matrices, and they're all the chain maps. And now I'd like to tell you that from this algebraic topology, I can tell you a lot about dynamics. These are now theorems about dynamics. Okay, so let's focus on this. That's supposed to be a hysteretic phenomenon. Can I see that from the algebra? Well, there's my starting state. I pour in the S and I get moved across. The algebra here is telling me that this generator got mapped to that generator. That's what the algebra says. Well, if I think about that from a point of view of a hysteresis diagram, I don't know that I'm talking about fixed points or anything like that, but if I think about it from a hysteresis diagram, I'm starting at low S there, and as I map forward, I follow a branch. It's an inclusion map. Well, let's go a little bit further. Gee, I ended up someplace else. If I do the algebra, well, I do have an isomorphism from here to here, but it's definitely not an inclusion map. That's what you would have expected to see from the dynamics if you kept tuning this S. Hysteresis is, if you go back, you don't end up where you started. Let's do the algebra. You go back, that's the algebra here, and if you do the algebra, you don't end up with the same place that you started, because you go back through an identity, an inclusion map. So from the algebra alone, I can start seeing hysteretic phenomena. And these are theorems now, right? Any time you have this kind of transversality structure, we get these results. Let's exploit this a little bit more. If I give you Hill functions now, which is what a lot of people like to model, and we modulo a small detail, if you set D equal to infinity, we can prove, I can't claim a proof, there's a technical thing we still need to overcome. I just don't think I've had enough time to write it down, all right? So as confident as I am without having written a proof, I am confident that using this, if we put Hill functions, well, for this I know it's true, but for arbitrary Hill function models, if you make D sufficiently large, then this computation is going to describe the global dynamics of the Hill functions everywhere. Right? And it's going to capture all the equilibrium structure and more. But let's do even more. Let's start here and let's run around in this direction. The algebra tells me this equilibrium changes to that equilibrium, includes into that equilibrium. Let's go around this way. That says this equilibrium goes to that equilibrium, includes into that equilibrium. I don't have equality. Right? So I have a diagram that comes around and I don't have equality, but this is a contractible space. So there must be some kind of singularity going on there. Now, this is simple enough that I can put something down here. I can put in a cusp bifurcation, and that's exactly what you would expect to see if there's a cusp bifurcation sitting there. The reason I get excited about that is that cusp bifurcations are not removable singularities. So this says that if you perturb whatever system I'm working with, this is still going to be there. This makes me think that maybe this global structure is actually very, very robust. And the only way you can get rid of this object is actually the fact that there's another cusp singularity here, and so you have to change parameters enough so that these cusp singularities cancel each other out. Okay, this is, I mean, I would like to be able to state this conjecture as a mathematical statement. This is the best I can do at the moment. It's robust. I don't know what robust means, all right? Obviously, the parameter values change as you change the D's that correspond to the boundaries, but at least for the toggle switch, I can tell you where they are if you do piecewise linear functions. So I would like to understand what it is that I'm looking at when I look at this level of dynamics and what robustness means. Right? So our numerical experiments show that this kind of structure exists for a much smaller D than anything close to infinity. Clearly a lot of work here, and most of it is done in joint work with other people. It means that most of the work was actually done by others. Sean Harker was a person who put together the software, the database software in the first place. Marcio Gamero has taken it over, and in particular is developing the homology aspects to it. Shane Kepley is a postdoc in Lunzang, and they have done this work on identifying the parameter graphs. This is a hard problem in computational algebraic geometry. Kelly has been doing the work on the connection matrices. The biological application and the idea of the whole approach is due with Thomas Gideon and Bree Cummings at Montana State. The attempt to prove that this is really true for all Hill functions being done with Tomas, Hiroshi, and Hiroe. And this idea that we should look at dynamics from the point of view of closed sets and lattices, that's due to Bill Cleese and Rob van der Horst. And I've run over by quite a bit. Sorry. Thank you.
Even though we're over, let's take time for a question or two. I had one.
What's a Hill function?
Oh, it's just putting in these nonlinearities.
I mean, who's Hill?
Looks like a hill.
That is the standard tool in biology.
Hill was a biochemist.
Yeah, sorry. Yeah. So here's my L's and my deltas, and you can see if D goes to infinity at this point.
So Jim Sethna
Yeah.
has a whole theory about sloppy models
Right.
that allows you to, in models that do have equations, that identify parameters that make a lot of difference versus ones that make a small difference. Do you think you can connect?
Shane has been telling me that that's what we need to think about. So Shane Kepley. So yes, that's definitely on our radar. If this is on my part a time constraint, but I think it's a really interesting idea. If anybody would be interested in doing that, who knows? If you want to talk about that, let's talk.
Do you have any comments about that, Jim?
Yeah, I missed the first 10 minutes, and I've been a little lost since then, but it seems as if there's some part of this, those flows have to do with the transience and long time behavior, and it seems like
So let me step back.
Yep.
So when I think about this picture here, this transversality model, right? What I'm really thinking is that I have no idea what's happening in this part. I'm assuming that somehow along these faces, I can tell you which way the vector field is.
Yep.
That's why I keep making this big deal about don't get hung up on this picture, because if I took the abstract picture and I put it in there with wiggles, as long as I can check for transversality, everything I've told you is correct, because all the theoretical results come from the algebraic topological.
So even though the upper right-hand corner has flows outward in both directions, doesn't mean that it's transient. It could have a attractor in it.
It could have a attractor in it. So, you know, when we actually went and I gave you something that was kind of consistent, what I'm really telling you is that if I get to this boundary, I have to move across. Okay? That's all I'm saying. And so this is kind of a very, in here, I'm going to assume that there isn't any recurrence. Why? Because I'm thinking of it as a perturbation from
Yeah,
what's
no,
going here. Right?
I I get that.
And in here, I haven't, that's why,
I'm pausing, trying to make some connection to the stuff we do.
Okay.
And we tend to deal with systems where as we change parameters, very little happens. That the reason that the behavior is simple, even though you have many parameters, when you sweep through all the parameters going from -a to infinity, it tends to do something boring, not new qualitative things.
right, I mean,
And
this is basically saying this chunk of 8-dimensional parameter space, at least to this coarse approximation, I don't see any difference, right?
yeah,
I mean, the actual attractors, if you start to give me funky nonlinearities,
Yeah.
they might be undergoing a lot of changes, right?
Yeah.
But in terms of saying you're gonna end up high green, low red,
yep, 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 yep. Mhm.
So I just want to get this straight. In the practical nature of this, the goal, which I think you mentioned briefly, is not to reduce yourself down to a model with we can give predictions. It's more of an exclusion thing where you're trying to do a process of elimination to eliminate all the other results that maybe won't hold for this. And then you can inform the experimentalist or whoever you're working with that, you know, this class of models is actually, this class of parameter space is actually, you know, you can hang out anywhere in here, and that's probably a decent approximation, and that's the most likely thing given that, I don't know, it seemed like you only had a couple assumptions on something goes this way or turns off, turns on.
so yes, I agree with what you said, except that I think we can do even a little bit more. So, and this goes to this very last statement about robustness. And I would love to understand what I'm talking about when I say that. Right? So I can only give you instantiations. So Lunzang has been working with Staj Schwartzman at Princeton. And Staj works on developmental biology, in particular the oocyte, am I pronouncing it right, for the fruit fly, all right? That model shouldn't fit into what we're doing. When Lund kind of started just throwing this machinery at it, I was kind of like, oh my God. Okay, so what Stas is really interested in for biological reasons, he wants to see a symmetry breaking bifurcation in his model. And well, we can run this, and then you find out where you have the symmetry breaking phenomenon. And again, there's all these step functions, and so, you know, Stas was kind of, well, I don't care about things out with these hard step functions. So what they did was they wrote down the differential equations that Stas is okay with, Hill function type models, and chose small d, and then went to the parameter regimes and randomly sampled from the parameter regimes and then put them in the differential equation and asked, is the symmetry broken? And it turned out, and this was just told to me on Wednesday, so I haven't even seen the data, it's just Lund told me this, that in one of these regions, 80% of the parameters gave symmetry breaking, right? It obviously can't be exactly the same. I mean, these semi-algebraic sets were gotten for very specific polynomials, and we've now moved away from them. So these regions that separate, well, they're going to start wiggling around, but somehow they didn't wiggle that much that we couldn't find substantial examples of the symmetry breaking. So there is something more going on. I just don't know how to say it mathematically.
Okay, well, let's thank
Thank you.